So what we end up with after oxidative phosphorylation is we end up with an average yield per substrate molecule. We get about 2.5 ATP per NADH, and we end up getting about 1.5 ATP for FADH2. Okay? If we were to add that in or multiply out based on the number of NADH and FADH2 molecules we had after glycolysis and the citric acid cycle, this would give us about 28 to 30 ATP from oxidative phosphorylation. Add this to the substrate level ATP that we got from our different processes, and we end up with our total yield per glucose molecule. All right? So that takes us through this process of ATP synthesis. Again, I'm not that concerned with the intermediaries of glycolysis or the citric acid cycle or anything like that. I really want you to be focusing on what's happening, and that's the fact that it's just a big series of redox reactions pulling electrons off of those fuel molecules so that we produce NADH and FADH2. Those ultimately go through oxidative phosphorylation, and that's how we make ATP. If oxygen is not present to accept the electron at the end of the electron transport system, the process can't continue. If mitochondria aren't present, well, obviously this is where we find the enzymes for this process, and the process can't happen. And that's why aerobic respiration requires both oxygen and mitochondria. Okay, so what we've done is we've followed a glucose molecule through this entire process, but it's important to remember that other fuels are important as well when it comes to aerobic respiration. Let's go ahead and talk about some of these other fuels and how they're metabolized here really quickly. The first one that I want to cover is just simply glycogen. It's our other big carbohydrate. It's an important one. So let's go ahead and talk about how glycogen is metabolized. You remember that glycogen is just simply a string of glucose molecules, right? So we have these 6 carbon sugars that are sort of all strung together, right? Oops. Something like this. All right, so on and so forth. Glycogen is obviously a lot bigger than this, but this is what we have. All right. So what we would normally do is, or I guess the logical thing to do, is if I were to ask you how do you metabolize glycogen, you would say that what we're doing is we're cleaving off individual glucose molecules and sending them into the pathway, right? And that isn't entirely wrong. We do metabolize some of our glycogen this way. However, there's one thing that's worth mentioning, right? So here's our glucose. And we've talked extensively at this point about how glucose enters anaerobic glycolysis. And I said that I wasn't in that focused on the intermediaries, but there is one that I want to mention here really quickly, and it happens to be the first step of glycolysis. In the first step of glycolysis, glucose is converted into what we call glucose 6-phosphate. All right? This is glucose 6-phosphate. Okay. Simply what we've done here is we've added a phosphate to the 6th carbon on glucose. All right. Where do you think that that phosphate comes from? It comes from ATP. Right? So we're using an ATP in the first step of glycolysis to make glucose 6-phosphate. Right? Remember we mentioned that there are some ATP being used. This is one of them. So the first step of glycolysis, the conversion of glucose to glucose 6-phosphate, is an ATP consuming step. Well, if we do this enough, that ATP adds up. Right? So when we break down glycogen, there is another metabolic pathway that allows us to bypass that conversion to glucose. There's another pathway that allows us to go directly to glucose 6-phosphate and bypass glucose. That saves ATP. Okay? So about 90% of glycogen is broken down directly into glucose 6-phosphate, right? And this is an ATP saving step. There's obviously a little bit more to this. I'm just giving you a really brief overview. We're not going to go into the detail with this as we did with glucose, but I do want you to be aware that there is a little bit of a kind of biochemical trickery going on here that allows us to save some ATP. All right? Okay, so that covers carbohydrates pretty well. All right, and then of course glucose 6-phosphate would just continue through glycolysis, the citric acid cycle like we've discussed. What about other fuels? Let's go ahead and talk about proteins. We know that proteins are long chains of amino acids, and before they can be metabolized, they have to be broken down into the individual amino acids. Right? They have to be broken down into their individual amino acids. We don't normally metabolize protein. We will metabolize a little bit of dietary proteins, but unlike fats or carbohydrates, during long-term exercise, we don't typically rely on protein stores for fuel. And if you think about it, the majority of your protein store is muscle. And you really don't want to start breaking down muscle for fuel. The only time you might do that is during starvation or something like that. But we don't normally break down protein stores as fuel. But we will metabolize some dietary protein, and so it's worth mentioning this. Okay, so our amino acid, if you remember, looks like this. Okay, we have our carboxyl group, COOH. Okay, we have attached to a carbon, we have our amino group. Okay, so that's what makes it an amino acid. An additional hydrogen here, and then we have our variable R group right here, right? So that R group is going to cause these guys to sort of vary a little bit in how they're metabolized. But the one thing that they do hold in common is this very first step, all right? The first step that we have here is called deamination. And in deamination, what we're going to do is we're going to pull the amino group off of our amino acid. Okay, so we're going to then have our let me see here. I want to make sure I don't screw this up. All right, we end up with something that looks like that. Okay, this is an organic acid. right? There's our acid, our COOH, our carboxyl group, and then we end up with that amino group that gets cleaved off. That amino group, that NH2, is almost immediately going to get converted into NH3 or ammonium. Okay. There is some oxidation here. This is an oxidative reduction reaction, so we are also producing a high energy electron carrier during this process. Okay. So this is what the process of deamination looks like, and this is going to take place regardless of the amino acid that we're dealing with. We're always going to get an organic acid, and we're going to get ammonia as a result of this. Now, because that R group on the amino acid is variable, the organic acids can vary quite a bit, right? So those organic acids can either be directly or be converted to pyruvate. They can get converted to acetyl-CoA very easily. Acetyl-CoA very easily, or some intermediary of the citric acid cycle, right? Citrate, etc. The bottom line is that all of these guys are going to eventually produce ATP, right? If we start with pyruvate, well, we just went over that pathway. If we start with acetyl-CoA, you can start the pathway that we discussed at acetyl-CoA. If we start with citric acid, you can start there, so on and so forth. So depending on the amino acid that we're dealing with, the ATP yield is going to be variable. So this is variable ATP yield, right? The organic acids that are converted to pyruvate are obviously going to be yielding more ATP than the ones that are converted to citrate, so on and so forth. So it just really depends. All right. The other thing that we do have to deal with when we metabolize protein is we do have to deal with that ammonia that's being produced. As you're probably aware, ammonia is extraordinarily toxic, right? So what happens is that ammonia is going to be detoxified fairly quickly. It's going to pick up a proton and get converted to ammonium very quickly. It's going to get converted to ammonium very quickly. Ammonium is a lot less toxic. That ammonium is eventually going to get converted to urea, and the urea is going to be excreted in our urine. Okay? And we're going to talk a lot more about nitrogen metabolism later on in the semester. But for now, just know that we have to get rid of this toxic byproduct, or else we can run into some real problems. This whole process is one where we are putting a lot of stress on both our liver and our kidneys. And we often see in people who have like sole protein diets or super high protein diets, they do have slightly more stressed kidneys and livers than the average person, because this is a fairly stressful process that has to take place in order to get rid of this toxic byproduct of protein metabolism. All right. So with proteins, we are getting fairly variable ATP yield depending on the individual amino acid that we're dealing with. All right, what about lipids, specifically fat? Okay, what about fat? Now remember that our fat molecule is a triglyceride, right? So we have our glycerol, and we have 1, 2, 3 fatty acid chains. I'm not going to write it out, we're just going to say fatty acids, right? 1, 2, 3 fatty acids. Now those fatty acids, okay, are these big hydrocarbon chains. these hydrogens in really quickly. So on and so forth, right? That's what one of those fatty acids looks like. This one's probably a bit short. The ones that we are metabolizing are typically a little bit longer, but you get the idea, right? So the first thing that has to happen is that we have to cleave, when we metabolize a fat, we have to cleave the fatty acids from the glycerol. So the first thing that we see is the cleavage of the fatty acids from the glycerol, okay? That glycerol is going to be metabolized, okay? And we're going to get some ATP from glycerol. But the real money comes when we look at the fatty acids themselves, all right? The fatty acids are going to be transported into the mitochondria, okay? You guys are going to be reading a paper by Raul Suarez that talks about the transport of the fatty acids into the mitochondrion. And once they're transported into the mitochondrion, they're going to be metabolized by a process called beta oxidation. Okay? Beta oxidation is a process by which we're cleaving off two carbon groups. So we're doing this, we're doing this, we're doing this. Okay? Those two carbon groups that we're producing as a result of this, these two carbon molecules that we're producing, are getting converted to acetyl. Why am I having such a difficult time writing? Acetyl-CoA, right? So as a result of beta oxidation, we're producing a ton of acetyl-CoA. Well, what does acetyl-CoA do? Acetyl-CoA in the mitochondria combines with oxaloacetate to make citrate. And we have a turn of the wheel, right? We have the citric acid cycle happening once for every single one of these acetyl-CoA molecules that we produce. We saw in the last lecture, every single time we go through the citric acid cycle, we're producing a ton of NADH and FADH2. And that then goes on to make ATP in oxidative phosphorylation. So in this hypothetical fatty acid that I've drawn, we're producing 1, 2, 3, 4 molecules of acetyl-CoA via beta oxidation. So this single fatty acid would turn the citric acid cycle 4 times. That's twice as much as a glucose molecule. If we look at the fact that we have 2 other fatty acids on this fat molecule, what we would see is that we wouldn't be turning it 4 times, we'd be turning it 1, 2, 3, 4 times 1, 2, 3. That's a total of 12 times. So as a result of that, the ATP yield from fats is typically significantly greater than we see for sugars or proteins. This is why when you look at the side of a cereal box or something like that, and it talks about different things like fats and proteins and carbohydrates, and it talks about the calorie yield, all you have to do is get out any box, look at the nutrition information, and you'll see that fats have more than twice as much energy than proteins, and almost twice as much energy as carbohydrates per gram, right? But this is because the fatty acids are essentially turning that wheel of the citric acid cycle so many times. So fats are just a really, really energy dense fuel source. Fats are incredibly energy dense, right? those numbers that I mentioned just a second ago have 9 kilocalories per gram. Carbohydrates, I misspoke slightly, have 4 kilocalories per gram. So fats have more than twice as much energy per unit mass than carbohydrates do. And it's because of their structure and how many times they turn the wheel. All right. This has a lot to do with the fact that, or this is the reason why you run primarily on fat. Right? Sitting here right now, watching this lecture on your computer, or on your phone, or what have you, assuming that you're not at the gym or something like that, multitasking, you're running primarily on fat. All right? At low metabolic rate, you tend to run on fats. Okay, so at rest or low intensity, okay. At rest or low intensity, you tend to get most of your energy from fats, and you get a very small amount from carbohydrates. All right. So sitting here right now, you're running primarily on fats. Walk to your car after class. It's still fairly low intensity exercise. You're running primarily on fat. And the reason behind that is because you get so much energy from it. All right. However, when you start to exercise, and as you exercise at higher intensity, the pattern shifts. We have fats, we have carbs, and we see the pattern starting to shift. We see that we rely less on fats and more on carbohydrates to fuel our exercise as we start to exercise more intensely. All right? So there's a pretty significant difference here in terms of the way that we utilize fuels at rest or at exercise. And again, the paper that we are going to be reading by Raul Suarez has a lot to say about this. All right. This is why when you want to burn fat and you want to go to the gym, you're not going to run on the treadmill as fast as you can possibly go and exercise at very high intensity. It's because your body shuts off its fat pathways and it starts to rely on carbohydrates. If you want to burn fat, go for a walk or go for a jog at fairly low intensity. That is going to help you to burn fat. But if you want to start depleting carbohydrate stores, well, that's when you'd want to exercise at a very high intensity. All right? And as you're probably aware, you don't store as many carbohydrates as you do fats. So as a result of that, you're going to run out of fuel faster and you're not going to be able to exercise for as long. But if you can maintain that reliance on fats by exercising at a low intensity, you have great fat stores and you're going to be able to exercise for longer as a result of that. All right? Okay. One other thing that I want to look at with this, we know that fats are more energy dense. All right? However, we notice that we don't get as much energy from them all the time. So let's go ahead and kind of look at some other numbers from some empirical data. If we look at lipids, or fats, and we compare them to carbohydrates, one of the things that we see is that fats give us 5.6 moles of ATP per mole of oxygen that we consume. Carbohydrates give us 6.2 moles of ATP per mole of oxygen consumed. So the energy yield per oxygen consumed is actually greater for carbohydrates. All right? Fats have more energy stored in them, but we're actually getting more energy from carbohydrates. And the reasoning behind this is not very well known. We know that this happens. It happens more in some tissues than others. The heart is a really big culprit of this. where you actually see a bigger discrepancy even than this. This is in about an 11% difference. But in the heart, we see that carbohydrates give us about 30% more ATP per mole of oxygen consumed. And again, the reasons behind this are not very well known, but we know that when we metabolize fats, we are wasting some oxygen, so to speak. It's called the oxygen wasting effect of fats. And there are multiple hypotheses kind of floating around this, or about this, but it's hypothesized that as we metabolize more fat, we're actually incorporating more unsaturated fats into our mitochondrial membrane. And what that does is it allows protons to leak back through the mitochondrial membrane and not pass through ATP synthase. When fats leak back through the mitochondrial membrane and don't pass through ATP synthase, they aren't producing ATP. And what we're doing essentially is we're uncoupling oxidative phosphorylation. The electron transport system is getting uncoupled from ATP synthase, and it's hypothesized that fats kind of enhance this uncoupling effect. And that's kind of the current hypothesis. But we don't know for sure. It turns out that there are lots of other things that uncouple ATP synthase from the electron transport chain. Cyanide is a poison that does that. There's a rat poison called rotenone that also uncouples the electron transport system from ATP synthase. Oligomycin is a really popular antifungal medicine that is an uncoupler as well. So there are lots and lots and lots of things that uncouple these processes from one another. All right? Okay, so that's a little bit about different fuels and how they're metabolized and things like that. We're going to be looking at this in lecture in a little bit more detail. Now, I want to talk really briefly about synthetic pathways, right? We've talked now about how things are broken down. Now we want to talk about how things are made.